Welcome back, everyone, to the OIS Retina podcast program. This is Firas Rahhal. I am a partner here at LA Retina, as it's known, or Retina Vitreous Associates, and also a general partner at Excite Ventures in New York City. And I'm delighted to have a good friend as my guest today, Wesley Jackson, PhD, as opposed to some of our other doctors who are clinicians. We'll hear more about that later. Chief Scientific Officer and a board member for Valitor, a company that's working on very interesting drug delivery products for the posterior segment of the eye. So it's highly relevant to this audience. Wes, thanks for coming on. I appreciate you taking the time and educating us a little bit today about drug delivery.
Yeah, of course. I've tuned into several of these and excited about the opportunity.
Join Doctors Maria Barakal, Praveen Dugal, and Firas Rahhal at the upcoming OIS Retina on July 27th in Seattle, where you'll see and meet with leading startup companies, top industry executives, and clinical thought leaders addressing unmet retina needs through novel therapies. Save 30% when you register for both OIS Retina and OIS 13, the original Ophthalmology Innovation Summit, taking place this December 1st and 2nd in San Diego. Register today at ois.net.
Thank you. And truth be told, we know Wes very well at Excite because we've been working with him for a long time, and then ultimately did make an investment. So full facts on the table into Valitor. We're super excited about it. We're super excited about him and the other members of his team, who I'll give him the opportunity to tell us about. But let's start. I always like to start with some background on you personally, Wes. I've gotten to know you over the last couple of years. The audience, some of them may know you, some may not. Your educational background is you have a BS in bioengineering from Berkeley, also an MS in bioengineering from UC San Francisco, which is, of course, the medical school and graduate schools only, and a PhD in bioengineering from Berkeley and UCSF. That's an interesting combination. I'm going to want to hear about that. But then you also became a scientist at Berkeley after your training, writing grants, doing research. What can you tell us about these early educational and early employment opportunities for you?
Yeah. So just to address the joint program between UC Berkeley and UCSF, I think the bioengineering department, as they were developing the grad school here, really wanted to take advantage of the engineering resources at UC Berkeley, as well as the really strong medical resources at UCSF. So as they put that program together, it really was a joint program where classes were taken on both sides of the bay. It was a little challenging coordinating schedule sometimes, but then we had access to faculty both at the medical center as well as in the School of Engineering. So it was a really great way to kind of merge the expertise in the two schools. And through that, I'd really developed a strong interest in biomaterials and the way biomaterials were able to be designed in a way that they could help better deliver drugs, better instruct the biology in the body to react to devices, implants, other drugs. And so that was really the focus of my PhD there. Before I came back to work at UC Berkeley, I went to the National Institutes of Health and I worked as a postdoc researcher there. Again, using biomaterials to develop tissue engineering constructs, understand how cells reacted to the biomaterials, to the cytokines that we were also treating them with to develop different types of implants and regenerative medicine strategies. And it's really while I was there that I had started to really appreciate the strength of the biological components that we were putting into these constructs that we were trying to engineer. So I started seeing my interest shift away from the material science and more towards the biotechnology development components of what we were doing. So as I moved back to Berkeley, and was looking for opportunities to get involved in biotechnology, I actually first reached out to some of my faculty advisors at UC Berkeley, Kevin Healy and David Schaefer, who had the technology incubating in their labs that I would eventually help spin out into Valitor, and would become the foundation of everything we're working on at Valitor. So the interest in the Valitor technology came first. We decided to put a company together, and then a little bit of the onus was on me then to figure out, as I was in these early stages of company creation, I needed to be able to work pretty much full-time in the company if any of the grants we were writing were to become achieved. And so it was during that time I was doing a lot of work, grant writing. I had a part-time position at the university, again, helping some of the academic researchers there write grants, write manuscripts. And so it was all part of a strategy to launch Valitor, and along the way be developing a lot of the skills around grant writing, telling the story of our technology, and eventually working towards building Valitor as a full-time job for me, and the ability to really start growing the company.
So it sounds like it was your intent to do this type of work to ultimately go into the private sector and spin out a company. Sometimes it's the other way around, and people are going in with an academic career in mind and decide otherwise somewhere along the line. Was that your plan all along? And then if you could, you wrote grant applications in so many areas, I'm sure. Multivalent conjugate drug development, that's a term. And I'd like, after you tell us a little bit about your original intentions there, maybe define that for us. Tell us what that means, because I know that's going to lead into our discussion about what you're doing at Valitor.
Sure. And I think I've had some pretty sharp U-turns in my career and intentions over my lifetime. I think I was really lucky early on in my academic career as an undergraduate. Actually, I had the opportunity to work at a medtech startup through a very random connection. My dad's college roommate's sister's brother-in-law was a very successful medtech developer, had exited and was starting a new company. When I was hired, I was the 5th person hired as a summer intern. And by the time I got there, there were eight people working. So it was really in a growth phase. And so I spent the summer in a startup, a very small startup, really falling in love with that type of environment. It was fast-paced, decisions were made quickly. And it was really fun to see, from the CEO down to the intern, everyone kind of pulled in the same direction, and it was all hands on solving whatever needed to be solved on a daily basis. So I actually got to go back to that internship a second summer, and it had grown from 8 to 10, where I'd left it the previous year, to like 30 to 35, just over the course of the 9 months that I was gone. And so I experienced kind of a different phase of a startup, and in some ways it was a little bit more corporate, but there was still this like, we're all in this together. Everybody is doing whatever it takes in order to make sure we get this product out in a timely fashion. And so the startup culture just kind of got in my blood at that point. And I had a lot of interest in being a part of the startup community from there on. But at the same time, as I was going into graduate school, my interests were really in an area that was much more biology focused, much closer to biotechnology. And so I put my ambitions to go into the startup world on pause a little bit, assuming that the types of work we were doing, I might need to go into academia and able to continue doing that. As I would be thinking in the back of my head, what would it take to actually fund a startup company doing some of the work I wanted to do? And it would just require millions of dollars of upfront costs of infrastructure in order to make a company happen. And while I was at the NIH doing postdoc work, the incubator facility here at the USCSF campus through the Quantitative Biosciences program was launched, and I heard about it, and it just, you know, rekindled this hope that we could start a startup company on a much lower budget, where instead of having to rent a whole facility, we could rent a single bench at a time if that's all we needed in the lab. There was no need to buy big pieces of capital equipment. We could be renting it by the hour if we needed it. So the pathway to start a company suddenly became realistic again. So as we moved back to California, my family moved back for a number of reasons, but one of them was to take advantage of the startup infrastructure for life science companies at the time. And so as I was going back and talking with faculty advisors, you know, it was kind of with the intention of hopefully finding a technology that was ripe for translation, for commercialization, and that we could spin that out for a startup company. And it was just fortuitous that I met up with two faculty advisors who I rotated through Kevin Healy's lab. I didn't spend a lot of time in his lab, but I learned a lot of important chemistry, a lot of important biomaterial science, and the underlying technology behind the multivalent conjugates that we develop at Valitor came out of that lab and out of a collaboration with his co-founder, David Schaefer. So, you know, from my perspective, it was a very exciting technology. I imagined a number of different avenues we could develop the technology and the different types of products. But it was also a great on-ramp for me to move into the biotechnology space because the core of the technology is material science. So, you know, it was an area that I had a lot of understanding, a lot of expertise that I could apply to a biotechnology product in order to make it a better drug. So that's how I ended up in the private sector startup space and specifically around the technology that would eventually become Valitor.
I'm glad you're talking about the early spin out of Valitor and how you know, at least conceived that. Take us through some of the actual technical process, if you don't mind, because this is of great value to young entrepreneurs or academicians who are thinking about it. What do you recall were among the early significant challenges? How did you view the tech transfer process? Were they helpful? Intellectual property challenges? Obviously, the financial challenges are part of all this. Please share with us. And who did you have helping you through that process at that time? Were there professional hires or just friends? How did it work? What was the process?
Well, I think I was really fortunate at the time as the QB3, the Qualitative Biosciences Institute, UCSF was developing the incubator system. They also had put together a really great seminar series that walked through all of the logistics of starting a business. So, you know, there was a, you know, hour-long seminar where you would go and learn how to register yourself with the government so that you could write a grant and have a grant funded through the NIH, which is not entirely trivial to do it the first time. And at least when I was doing it, the instructions on the internet were not great. So it was very easy to make mistakes.
You couldn't Google everything in the world in 2 seconds yet.
It was not as user friendly. You know, there were seminars on how to build a chart of accounts and how to do some basic bookkeeping so that you would have financial records for money coming in and out of your company. And, you know, as a primarily academic scientist before this, you know, we just sent our invoices off to an office. We didn't know what was happening behind the scenes. But when you're in charge of the company, you need to understand a little bit of accounting. You know, there were seminars on how to use your corporate counsel. What is your corporate lawyer for? And what can you be looking to that person to, you know, make sure you're in compliance for various different aspects of the business. So a lot of the foundational knowledge of just company creation I got through this seminar series, and fortunately they've kind of maintained that. So as new entrepreneurs are coming out of the university, they're able to access these types of seminars, which you know, continue to be the foundation of a lot of just the company logistics that are necessary to keep the company going.
You didn't have to hire and spend a lot of money to do some of these early steps because of the program. Do most universities have these types of programs?
I think it's becoming much more common now because there's a real strong drive for universities to see their technologies spin out from internal faculty members, postdocs, graduate students. I was actually in a cafe here in Berkeley, and I happened to sit down next to the Office of Technology Licensing group as they were having coffee. So this is maybe off the record, but the discussion they were having was the motivation for the faculty members or the students who are spinning the technology out of their own labs. There's just so much more ownership over seeing that technology through to a product that generates revenue, as opposed to licensing it into a larger company where it may get lost or fall between the cracks or nobody really takes ownership of it. So I found that to be an interesting little nugget that while sometimes it feels like the Office of Technology Licensing has systems in place to make them compatible with larger companies, their preference is actually to see it go towards whoever's going to have the most ownership of the technology, and often that is the small company that's being created out of the university.
For what it's worth, I agree with the concept, which is bigger companies may have a tendency to shelve something, and a smaller spin out, because it's their life's work, is going to run with it as long as possible. And for the record, I think that made it not private. We just unless we edit that segment out, I think you're on record now.
I think we're okay.
In your opinion, and I know this is opinion and not fact, so please share whatever you feel. When is the right time to move forward with a spin out? Let's say you're a postdoc or a junior faculty member at Berkeley or one of these great medical institutions. When is the right time? Is it when you can raise institutional money? Is it when you think it's appropriate to build a management team? Is it based on where the technology is? What's your take on this? I think that's a tough question for most early entrepreneurs, is when do I do this?
Yeah, and I don't think there is a right answer, but I think from my casual observations, the right time is when you've got someone who's ready to take that ownership and take on the responsibility of seeing the technology through, because it's a big commitment. It's a lot of work, and when you've got someone who's enthusiastic, really sees the potential, has a vision for where that technology is going to go. Everything else is going to fall into place because that person is going to make it happen. And is that majority of the time the scientists themselves, or can it be anyone but you? Let's say with this technology, can you transfer that enthusiasm and skill with your product that you worked on in the lab to an outside individual, or does it really have to be that person? Well, I think maybe Valitor is a good example of that, because the original team in the lab that developed the technology as it was eventually patented and out-licensed to Valitor moved in a different direction. The idea of this multivalent conjugation, the drug development route just wasn't aligned with where their career goals were. But I came in as the technology was ready to spin out, and I had the support of the academic co-founders who wanted to see it translate, and then we developed the vision to keep it moving. I think the early steps of translating a technology out of the university are challenging, particularly if you're not the ones who had your hands on the technology all the way from the beginning. So, I think as we funded our first grant from the NIH, a lot of the time we spent under that grant was reproducing what had been done in the academic lab. So I think that just instilled a certain level of robustness to the technology, because we already had to replicate what had been done in the academic lab, make it work in our hands outside of the university. And then we were able to build a knowledge base around the technology that was independent of what was in the university before it spun out. So I think that helped us give us a really strong footing to build the company from there.
I got it. This is a good segue because I want to move to the current and talk a little bit about the technology. That's what, you're primarily here to tell us about. I'm always fascinated by the history, honestly, if not the same, more than the current, because everybody comes to these points in their professional lives through a different route, including yourself. That was very interesting. Take us through a little bit, either broadly or specifically, depending what you think is most helpful and useful, about this protein biopolymer conjugation technology that you're working on. And I saw that it's being referred to, at least online, as the MVP program. And we all know what an MVP is in sports. What does it stand for? And tell us about the technology in a broader, specific way.
Sure. So MVP at Valitor stands for multivalent polymer conjugates. And the way it originally was conceived was in one of my co-founders, Kevin Healy's lab, and touching a little bit on the work I did with him as a graduate student. The focus of his lab was what he referred to as cell instructive surfaces. So for implants in particular, as they are encountered by the immune system or other cells in the body, the goal was to make it so that it was more biocompatible. And one of the strategies that they used was using biopolymers to attach peptides or different types of cytokines to provide instructions to the cells who are encountering that surface. And that was a major focus of his lab around the time David Schaefer joined the faculty at UC Berkeley. And Dave Schaefer's lab was really heavily focused on the development and differentiation of neurogenic stem cells. And they can be quite challenging to maintain, challenging to differentiate. And so he was working with Kevin originally to see if they could develop surfaces that would help provide that communication to the stem cells in order to get them to differentiate or be maintained in the way they wanted the cells to grow. And somewhere along the line, they got the idea, well, what if we just detach these biopolymers from the surface and allow them to be freely soluble in solution? And they found that there was quite an enhancement in the potency of the drugs or the cytokines, the proteins, when they were attached with multiple copies along one of these biopolymers. And so, kind of the aha moment, at least on the academic side, is when they realized as you change the number of drugs along the backbone biopolymer, you could change the potency, and in fact, generate constructs that are quite a bit more potent than the individual drug molecules by themselves. And so that was really interesting as an in vitro tool. And so as we were forming Valitor, the question was, well, if we have this ability to enhance the potency of these protein biologic drugs, where would it make sense to use them in a therapeutic context? As we started doing more work with the MVPs, we started realizing the potency effect is very important. It's great that we can have a very long biopolymer, decorate it with multiple copies of the drug, and still maintain a really high bioactivity. But also the size of these MVPs is quite a bit larger than the individual drug components. So we can use that in order to engineer the PK properties of the drug in a way that can also improve the therapeutic efficacy of whatever drug we're trying to design. So in the spin out, once we'd formed Valitor, I think the big aha moment we had was that we could independently control the size of the biopolymer and the copy number of the drug on the backbone in order to independently control the PK and the PD properties. So that really kind of changed our outlook on the types of indications that we could look at in order to provide better medicines in that space that better match the needs of those clinical indications.
So you came to the eye, or the posterior segment of the eye for sure, hence where I come in, and this is the OIS retina program.
Mm-hmm.
Before we get into that, where else? Are there other areas that you're clinically interested in?
Yeah, so one area that I have a historic background in is orthopedics, and trying to understand how to reverse some of the damage that can occur in osteoarthritis, rheumatoid arthritis, a variety of different types of joint diseases has been an interest of mine. And we have a platform that enables us to provide long durability in intra-articular joint spaces as well. So we can deliver anti-inflammatories, we can deliver a variety of cytokines or anti-interleukin therapies that could target a variety of different joint diseases. And then we have a number of different strategies where we're going to apply again, a different biopolymer backbone platform, but going after a variety of different anti-cancer mechanisms. And those are much less developed internally, but continue to be an area of interest. And we enjoy seeing how we can take the basic principles of the MVPs and be applying those to generate, improvements in therapies kind of across the body.
So oftentimes, and I don't know this, even though we're involved with your company, I don't have any idea, so I'm open to the answer. Oftentimes, the back of the eye kind of can serve as a proxy for neuro or neurodegenerative processes. Is that an area also of clinical relevance to this program?
Possibly. I think we have some strategies of enhancing therapeutic activity beyond the blood-brain barrier that has been of interest, but a little bit further behind even the joint disease and
okay, got it.
Areas.
So you have the multivalent complex and you attach molecules to it that have some drug effect. I'm breaking this down as simply as I can, that then has an effect. Can this be attached to any drug or compound, i.e. the API? Is it universally tunable? Like, what are the types of molecules you can attach to this?
Yeah, so as we were at early stages developing the technology, my intention was to try and make it as interchangeable as possible. So if you had different biopolymer chemistries, different biopolymer molecular weights, we could apply the same chemistry to those for conjugating the drug molecules. We have fairly standardized methods of conjugation. It's nothing esoteric. We wanted to stick with chemistries that the FDA has already been exposed to. And there's conjugation handles both on proteins, peptides, nucleotides, small molecules that enable us to be compatible kind of across a range of broad types of payloads. And we've generated MVPs using all of the above. I think we've got areas where we find sweet spots of certain sizes of proteins, at least, that enable us to really maximize the range of pharmacological properties we can achieve. But one of our strategies from the start was we wanted to have a platform that would be compatible with a partner's payload. So if there was a problem with the pharmacological properties that a partner was developing, we'd be able to merge it with our technology, generate a new composition that would solve those particular pain points. And we want to do that without having to re-engineer the MVP from the ground up every time.
So are you currently working using your products with, let's say, in-house novel molecule / APIs and or external partnered molecules? Do you have interest in both? Are you doing both?
So we've got some ongoing partnership activities that I am not able to talk about. But
I'm not going to go there.
The internal APIs that we are developing do span a broad range of different therapeutic types for different indications. And one of the principles we've had in prioritizing products in our pipeline is to at least at first go after clinically validated targets for indications that already have established patient populations and a pain point that we can solve. And there certainly are a wide variety of indications that fall into that category. Many of them maybe don't have the strongest clinical validation yet, but as we see products moving into phase 2, phase 3, that's kind of a trigger for us to start thinking about generating our own internal assets so that we can move quickly on, you know, the later stages of development for those products once there's a clinical signal to follow. A lot of the work we do to develop the assets can be done in-house relatively quickly and inexpensively, and then you know, once there's clinical validation, that would be a trigger to start escalating the effort and expense that we put behind those portions of our pipeline.
How complicated, and this may be a dumb question or not, is building these super molecules scientifically, this process by which you attach the API to your larger, you know, carrier, so-called carrier molecule? How hard is that to do? And then I want to talk to you a little about some of the specific ones you're doing right now.
Yeah. So, you know, as we spun it out of the university, you know, at first our intention was to not try and fix anything that wasn't broke. So we kept a lot of the chemistry consistent. As we got deeper into development and as we were expanding the number of payloads we were putting on the backbone, we started realizing there were a lot of subtleties in the chemistry that would impact the overall performance of the molecules. So over a period of 2 or 3 years, we really kind of stripped down the platform to, you know, a bare biopolymer backbone, and then rebuilt the chemistry in a way that was optimized for the development of biopharmaceutical products. So, you know, the initial generation of conjugation handles on the backbone has some subtlety to it, so that we're not overly impacting the chemistry on the backbone that can cause problems and stability in vivo down the road. The choice of chemical linkers for conjugation have a big impact on overall performance. And then, you know, we've found some ways to provide better conjugation between the protein and the biopolymer, again, in a way that provides for improved stability, easier manufacturing that fits in a little more smoothly with the infrastructure that already exists for drug development and manufacturing. And so, you know, as we've built the IP portfolio around the conjugates, it's become quite robust, covering a lot of very specific compositions that come together to make the overall MVP conjugate. That being said, you know, once we've got that chemistry worked out, the overall process of manufacturing it, while not trivial, is relatively straightforward, and we have the ability to piggyback on the manufacturing process for one conjugate and apply that to, you know, the other pipeline products.
That makes sense. One of your main MVP or multivalent polymers, one of your products, is something very familiar to ophthalmic surgeons, based in hyaluronic acid. I'll allow you to tell me in some detail what I mean by that and what you mean by that. We know it commercially as Helon, or a number of other things we've been putting in the eye and using in the eye for decades, and they're amazingly useful products in the capacity that we're using them now. Tell us about your version of this, how it's similar or different, and share with the audience something you and I have talked about in the past, is what happens to these molecules after we've used them in the eye. What's the intermediate and long-term processing of this molecule after we've used it in the anterior chamber of the eye, much the way you might want to use it?
Yeah, so we do use hyaluronic acid as the biopolymer backbone for our ophthalmic products. You know, the hyaluronic acid has some really great biophysical properties that enable it to form these really big hydrated structures, which is useful in the context of the back of the eye, because there's a microstructure in the vitreous humor that the hyaluronic acid will get entwined within, and its ability to move through that, you know, microstructural matrix that it defines the rate at which it's going to be cleared out of the back of the eye tissues. You know, it also is useful for us that hyaluronic acid is a major constituent in the back of the eye matrix, because there is a exclusion of the types of enzymes that might otherwise try and break down the hyaluronic acid, because it's important to keep it intact to keep the properties of the vitreous humor. So the ability for this kind of worm-like molecule to snake its way through that matrix defines the PK properties while it's in the back of the eye. And that's what gives us the really long half-life for the MVPs we use. Our lead product is VLTR 557. It is a hyaluronic acid backbone with about 100 copies of a single domain anti-VEGF antibody. So as that hyaluronic acid backbone is, kind of slowly working its way through the matrix, it's carrying along with it these anti-VEGF antibodies. We've had some recent PK data showing that as it approaches the retina, we're getting penetration into the retina and choroidal tissues, and about the concentration in the retina is about 1/3 of what we're seeing in the vitreous. So while it's still in those ocular tissues, we're seeing high concentrations of the conjugated anti-VEGF. But eventually these constructs will be cleared out of the eye, either through the front of the eye in the aqueous chamber, where it can meet up with the lymphatics and go into the blood compartment, or they go out through the back and into the choroidal vasculature, and again into the blood compartment that way. Once they hit the blood compartment, the concentration of hyaluronidases in the blood is really high. Hyaluronidase has signaling functions in the blood, so unless it's supposed to be there, the body wants to get rid of hyaluronic acid as quickly as possible. So the backbone ends up getting digested and metabolized, and our antibodies are small enough that they're cleared out through the kidneys. So as we have been able to generate assays that allow us to detect the concentration of our drug in the systemic compartments at low enough concentrations, we're seeing about a 10,000-fold lower concentration in the blood relative to the vitreous. So it does get cleared out quite rapidly once it leaves the eye tissues.
It's really brilliant. And I find myself thinking these questions and asking this question of many innovators with great, unique ideas. And as silly as it sounds, it's a great idea, right? So it's optically clear, it's well tolerated in the eye. We've been using it in the eye for decades. Why has no one thought of this before? No offense to you and your team, but this seems like, wow, of course, hyaluronic acid, it makes all the sense in the world. What was the challenge in it, if there was one, that kept this from becoming conceptualized before?
Well, I think it like many things, once you see it, it seems obvious, and it really
exactly
the direction you're coming from. So, we came at this already with the multivalent conjugation technology in place, and we knew the multivalent conjugation enhances the potency of the drugs that are conjugated. And so from our perspective, we could deliver a highly potent, large macromolecular drug to the back of the eye, and it seemed to check all the boxes that we needed to get the PK properties that we wanted. More traditionally, I think the strategy of using biopolymers to change the size of a drug and enable that drug to stay in the target tissues longer is something that's more traditionally done like the pegylation route, where you attach a biopolymer or multiple biopolymers onto a single protein in order to get the size of that drug large enough to have the PK properties that you want. And I think the challenge there is the more polymer you decorate that drug with, the more interference you generate that makes it harder for it to hit its target. So there's kind of a limit of how long you can make that biopolymer before you start losing all the potency of the drug, because it's just completely wrapped up in biopolymer. We came at this with a technology that decorated the biopolymer with the drug. So the longer we made the biopolymer, we just needed to add more drug to it in order to make sure we weren't not only losing bioactivity, but actually enhancing its potency. So, I think it was obvious to us as we approached the space, but we started from a different spot with a different view of the technology. So it was I would attribute more to luck than anything else.
I knew you were going to say that, and it's very humble of you. It's a brilliant, obviously brilliant idea. I put my money where my mouth is. I believe in this project.
We appreciate that.
I want to give you, yeah, thank you. You're welcome. I want to give you a chance to tell us about where you are in the science of the preclinical programs right now, and also share with us about the corporate development. I know you've had a lot of corporate development in the last 6 to 12 months. I've been, part of that with you. Please share with the audience where you are, both scientifically and on the corporate side.
Yeah, so you joined us on our Series B round, and as we were completing our Series A, our primary goal was to define a composition that would be ready to move into CMC scale up and produce material for a clinical trial and complete the clinical trial. As we were describing the status of our technology and composition to the Series B investors, there were a number of expensive preclinical studies that we wanted to do to really validate the composition, as well as move from our lab scale manufacturing to a scaled up manufacturing batch size that would be sufficient for clinical trials. So, our current status is having completed the sets of pilot studies for our IND enabling studies that are as close as we can to replicating the IND enabling studies without actually, going to that extent of expense and scope. So we were able to do these radio labeling PK studies, really finely define the concentration of the drug in other tissues throughout the body. We were able to measure the ocular tissues in earlier studies, but it was really in the last year we were able to see the systemic distribution, determine whether we were seeing bioaccumulation in any areas. And so that really, I think, queued us up in a good position to move into the IND enabling preclinical study for PK and PD. The other kind of key preclinical study we've done is around the preclinical safety. So we did a pilot study in non-human primates with repeat dosing, following them out for 125 days, and saw no remarkable findings, which is what we wanted to find. And then on the CMC side is probably where we've spent the lion's share of our effort over the last year and a half, taking our lab scale synthesis, transferring that to a process that can operate at a scale that's sufficient for clinical trials material, and then building in the control systems around the process with analytics that'll enable us to release the GLP batch and our clinical trials batch. So as you mentioned, corporate development, this has been a big growth year for Valitor, as we had a really solid science team. As we are taking on these CMC activities, we've been building out our development teams. We brought in a chief development officer. We just recently hired a VP of CMC. We've got a dedicated program manager, as well as some support team members around the development side that are helping manage the work that's being done at the CDMOs, the CMOs, as well as the work internally that's supporting those outside organizations.
And Steve Lowe joined as CEO, who has a lot of people in this audience will know Steve, long history in biopharma, and I guess with Genentech Roche, so familiar to the eye space.
Yeah, so Steve's been involved in biotechnology for decades, which is great, considering that until he joined, I think all of us had worked the longest on any product at Valitor. And his expertise in ophthalmology, he jokes, is that he has participated in the program for the most commonly used anti-VEGF product when he was on the Avastin program. But right. He was familiar with the ophthalmic space, and I think he got really excited about the technology, and the team of scientists we have here, I think were a big draw for him to get to work with the team we have. And I think
he's a heck of a nice guy.
He is, and I often find myself smiling as I am having discussions with him about strategy and human resources, you name it, and we're just very well aligned. So it was kind of a fortuitous match that we found somebody who would merge into the company so seamlessly. And the timing was really good because as we were moving into the development manufacturing stages of the lead product, it was uncharted territory for me. And so, it's hard for me to hire a VP of CMC when I don't have any experience in later stage CMC and in the directions we were going. So he had the longer view of drug development and had a better profile of the organizational structure that we were going to need, this year and for the next 10 years, and how to start laying that together now. And so it's been great getting to learn from him because he's got a lot of knowledge, and I get to sit next to him as he's making these decisions now. So I'm learning a lot.
What are your guys collectively, the company, next steps now? Let's say scientifically or however you want to describe it. What's next for you guys?
Yeah, so our primary corporate goal is we want to see the VLTR57 lead product go into clinical studies early 2025. So we're all hands on deck, making sure whatever is needed to be done in order to make that deadline is getting done. While that is our primary goal, we still don't want to lose sight of the fact that we have a platform in ophthalmology that we can be building on, and we have other areas outside of ophthalmology that can also be quite exciting. So one of my jobs is to figure out how and where we can use personnel resources to continue some early work on some of the other pipeline areas, and help us prioritize what may be the next product that's going to go into that CMC pipeline and down the IND route. And that may be in ophthalmology, it may be in oncology. I think that depends on the data we're able to generate and pain points that we're able to solve in these clinically validated areas.
So it's probably wild conjecture, but I'll ask you to do it anyway, because this is what I do. Early 2025 sounds like a very robust and a worthwhile goal and realistic goal. So where would that place us? I'm a clinician, as you know. When would a product like this, if everything goes perfectly, and we know that's not how science happens and drug development happens, but if everything went perfectly, when could a product like this be available for commercial use to a retina clinician? 2028?
2028 may be very tight. Probably 2029 is more likely. And that's me trying to remember timelines off the top of my head. So
yeah, no, I got it. And there's a lot of clinical trialists who would listen to this and probably be able to give us both a pretty distinct answer off of the 2025 number, but that's more than great enough. And I think part of what makes it difficult to predict is, I think in the anti-VEGF space, there have been clinical programs that have tried to skip steps and have not necessarily been to the advantage of the overall program. So I think one of the advantages we have is we're able to look at how previous clinical programs have been run, what steps we maybe are able to do efficiently and where we may want to spend a little bit more time, so that we're generating the data that is going to be convincing to the clinicians, and that we will have the ability to best market it to the clinicians. So I think there's a lot of still learning to be done in the space before we're able to really peg down what the long-term clinical program is going to look like. Yeah, I think that's really smart and realistic. I've been heard to say many times, both in financial circles, clinical circles, scientific circles, anti-VEGF drugs are a really high bar. So you got to do your homework and bring a really good product if you're going to match and exceed what we've seen with these great drugs. And I'm glad there's a lot of folks like you trying to find ways to extend the durability and the efficacy of these already great products with new additional products.
And I have to give credit to the management and development teams that we've brought in who are recognizing where the bar is in this space and making sure that at this stage, when we're just at early steps of developing the CMC campaign, that we're putting in the controls now that will serve us for when we're in phase 3 and beyond. And that again is why it's great to have expanded the team the way we did. I think, as I said, I'm learning a lot, and my comfort level about what we're doing now is very high as I see how that's going to mesh into activities that will be happening down the road.
Thank you. Thank you, Wes. This has been great, really educational for me. I know it's been educational for the listeners. Wes Jackson, Chief Scientific Officer of Valitor. Congratulations really on your progress so far, and good luck, and thanks for coming on.
Yeah, thanks. This has been great. Appreciate the opportunity.